Thanks, Lisa. We'll start again. Thanks, everybody, for joining today. This is the last core seminar of the semester, so we're grateful you all are joining us. Hopefully you notice we're recording, so please be mindful and mute your microphone or pause your camera if you're not actively engaged. My name is Andrew Bernard. I'm the director of the centralized cores, if we haven't met or engaged yet. And we've been hosting these seminars to spread awareness about the cores. So I'm going to turn it over to Jim Chambers in 1 minute. But as we go, if you have questions, please feel free to put them in the chat. We'll do our best to monitor and bring them up as needed. And if you have any general questions about the cores or anything at all, we should have plenty of time today to engage. So without belaboring it, Jim, take it away.
Thanks, Andrew. So my name is Jim Chambers. I recognize a lot of the names here. So as you know, I run the light microscope facility. The facility here at UMass Amherst is a Nikon Center of Excellence. And what that means is we get a lot of support, like seminars like this, from the company, and a lot of expertise from them to help us kind of take your imaging to the next level. In my facility, we have, I think at last count, 15 microscopes that range from, basic stereoscopes to wide field to multiple 7 confocals, super resolution, 2 photon. Basically, you name it, we have it almost. We're missing a couple things, but we have almost all the technology that's commercially available today. So today, Joe DiPietro is going to talk to us about automating your experiments. And as we were bantering before the seminar started, I'm one of the people who doesn't like sitting in front of a microscope 24/7. So I really like the ability to automate things so I can do other things at the same time while my data is being collected or while my data is being analyzed. So I'm going to turn it over to Joe. And Joe, I'll let you introduce yourself, and thanks for giving the seminar.
Yeah, Jim, thanks for having me. And it's always great to work with you and your core. My name is Joe DiPietro. I'm the Senior Applications Manager for the Northeast. And, the reason why, so Jim and I talked about a little bit about what we wanted the seminar to be about. And I really hope that what you get from this is a little bit more of an understanding of the breadth of actually what can be accomplished with Nikon microscopes in the core and the software that we use to interact with them. So without further ado, I'll get into it. And I think a lot of you are probably familiar, but when I talk about our software, Nikon software, I'm talking about NIS-Elements. So automating your experiments with NIS-Elements and some of the tools within it. I'll just start off at first with what a standard workflow looks like. A lot of you who are doing microscopy will be familiar with this. We'll get into a little bit of how that workflow can be scaled up to more high content imaging and automation. And then we're going to get into how and why you would do more custom acquisition and analysis. And that's when we're going to get into the things like jobs and GA3. These are terms that we use for interfaces within elements to do automated experiments and automated analysis. And then I want to use the end of the talk to get into some of the newest stuff that we're really focusing on a lot, and that's using artificial intelligence to really bring analysis to the next level and automation to the next level. So, Jim says he doesn't like sitting behind a microscope, but if you're sitting there behind a microscope stand, right, you have a bunch of questions. You're not just there to look at stuff. Typically, you have some questions. Where are things? How do things work? What's there? Why is something doing what it's doing? When is something that I'm looking at happening? When is a certain dynamic event happening? These are the kinds of questions that you want to answer. So how do we answer those questions? Well, we do an experiment and we use the microscope as a tool to do those experiments. Some of the things that the microscope lets you do take images over time, maybe capture volumes, three-dimensional images of your sample. Maybe you're looking at different probes, how much of these different probes there are, how do they interact, where are they located? Maybe you're looking at different conditions, different chemical conditions, drug, no drug, that sort of thing. Maybe you need a certain sample size in order to have enough power in your experiment to come to some kind of conclusion that's important, right? Well, those are your experiments. So what kind of system do you need to answer those questions? Well, it depends on the question that you're asking and the experiments that you need, but some of the components that we offer on our microscopes are things like motorized components, different imaging modalities, confocal, wide field. We have different versions of the same hardware that vary in speed. So for example, you might be able to do a Z-Stack with just a motorized microscope, but if you want to do really fast Z-Stacks, then you might need something that we call a piezo that sits on the microscope and allows you to do those Z-Stacks even faster. And of course, in order to be able to interact with all these things, you need something that's a powerful, flexible software that allows you to utilize these things to the extent that you need to. NIS-Elements offers all of these things. And so our microscopes also have all of these different types of modalities for you to choose from. So let's get a little bit more into how you would use them, right? Perhaps you have a sample with a lot of different colors. Well, we can use those motorized components and the software to take a multi-channel image. We talked about Z-Stacks already. As we know, biology isn't just two-dimensional. It's, our bodies and our cells are volumetric, right? And so if we want to accurately measure them, we need to be able to capture pictures of them in that dimension as well. You might have a big sample with lots of cells or a big tissue that have interesting events in multiple places. It can take a long time to sit there and move around and find them all. So being able to capture images in a lot of different places can be important too. It also can introduce bias. This is another important thing that people don't talk about with multi-point is if you're just, using the joystick and riding around, you're going to find things that you think are interesting, but it could introduce bias into your experiment, right? So being able to do this in an automated way, in a non-biased way, is important as well. Maybe you want to image your entire sample. We can stitch it together, right? And of course, we talked about this a little bit, taking images over time so that you can capture events that are dynamic and look at live samples and stuff like that. So how would you do this? Well, maybe you scan the cover slip. This is probably what a lot of you do all the time, right? You put your sample on, you scan around the cover slip or around the dish. Oh, there's a cell that I'm interested in. Well, I have to optimize it. I have to make sure that the intensities are right, determine what I want to image about this cell, capture the image. All right, now I have my image. Now I need to find another one. And another one, and another one, and another one, and another one. And as Jim knows, this takes a lot of time to do this. And as you all know, as folks who are doing experiments, this takes a lot of time. It takes a lot of personnel, it takes a lot of training. You have to sit there and find these things over and over again. Maybe it takes days or weeks to get the number of images that you need. You know, sometimes maybe the sample that you have has the capacity to produce more data, but the sample goes bad by the time you're able to collect all of that data because you spend so much time looking through it, finding the things that you need. This is really where automation comes in, and a really nice software package like NIS-Elements comes into play, because now you can offload some of that into a pre-built process that will do it for you. In other words, what if the microscope can do the work for you? Let's look at a couple different examples. We're going to start with simple examples, and then we're going to move up, and so that you can see the breadth of simple kinds of analysis that can be done with the existing software that you have, and more complex experiments that integrate analysis into it to produce a decision-making experiment. And you'll see what I mean when we get to that. So let's take this simple example. You have an image here with different types of seeds, or they could be cells, right? Whatever. Some of them are round, some of them are kind of this elongated shape. We have built-in tools that allow you to easily be able to segment objects based on shape. And on top of that, you can even have it find all of those objects that fall into one category and reorient them and show them all to you. So then you can select through them and decide which ones are interesting or important or whatever. Now, this is sort of after the fact, right? We took an image and we're segmenting based on shape after we have the image. But wouldn't it be a lot more useful if you could have the computer know what it is you're looking for before you start imaging, so that while the imaging is happening, you can immediately find those objects of interest? So here we have a bunch of cells labeled in DAPI. You can see some of them are in just a regular phase, and some of them are dividing. So what we could do is we could take this first image, which is representative of the rest of our slide, and we could tell a computer, hey, these are the types of things that I'm interested in. Then you can have the computer and the microscope scan the entire slide automatically. Until possibly you could do it for a number of different ways. Like you can do it so that it continues imaging until it counts the number of these types of cells that you're interested in. You can maybe have it mark all the locations of these images and then be able to go back to those particular structures of interest. It makes it a lot faster. It's easy to do because you just have to train it once. And it's also really unbiased and repeatable. It's always going to find the same things. If you were to, for example, give somebody this slide and tell them, go ahead and mark as many metaphase nuclei as you possibly can, I guarantee you that even if you gave it to the same person, the next day, They're going to find different ones. They're going to find a different number of them. It's going to take them a long time to do. In this case, if you put this slide on the next day and gave it the exact same parameters, it will find the exact same nuclei every time. That's really powerful when we're talking about repeatability and science and doing your science in a non-biased way, right? So in this case, this is what we mean when we say high content acquisition. Sure, you can acquire a high number of images and lots of data manually, but when we say high content, we really mean doing it in this automated way. More images, analysis on each image, repeatable results. So you're probably thinking to yourself, all right, well, that sounds great, but how do I do that? There's different ways. And I think one of the great things about NIS-Elements is that not only do we have built-in assays that we're going to, or really complicated types of analyses that are custom, but we also have a lot of built-in ones that people don't know about. And so I just want to introduce some of those to you because they're really easy to use. They're already made and they already work. So we have a graphic user interface for measuring cell motility. This is something that a lot of folks are interested in doing in the context of their broader work, right? Live dead. We have the ability to do that really easily. Cell proliferation, measuring intensity. We even have a wound healing assay. And of course, cell counting or object counting, something that a lot of folks want to be able to do and doesn't really require any more work on your part, except for knowing where to find this GUI in the software. And just getting a little sense of how it works and adjusting the parameters. But of course, you know, while a lot of these tools are easy to use and they're widely applicable to a lot of people, really you want to bring your work to the next level. What if you want to do, you know, something more? And we talk about this in the context of using your Microscope software as your development platform. So not only using the software to capture the images and use the tools that are available, but actually use the software itself to develop your experimental paradigm and your analysis. So let's discuss some examples of things that you might want to do in this context, right? One of the things that you might want to do is a conditional experiment. For example, images have data in them, right? Whether it's a marker, whether it's the part of the cell cycle that a cell is in, how big of a colony has been formed. But you'll notice as you start using microscopes more and doing more experiments, a lot of the images you take maybe aren't as valuable as others, right? Or some of the stuff in images isn't as valuable as others. You might be interested in rare events that only occur a few times out of thousands of cells. You might be interested in specific reactions that only happen a fraction of the time. But with high content acquisition, you can capture tons of images and find the interactions or the unique things that are interesting to you that are kind of rare, and find those things and then have the software make an intelligent decision. So for example, if you have cells that have been dividing and reach a certain density, then inject the drug. Normally, the old school way of doing this is you're sitting there, you're taking images, you're watching your cells, you're like, oh, there they are. They've grown enough. I'm going to stop the imaging. I'm going to go prepare my drug. I'm going to inject it. And then I'm going to continue imaging again, right? Well, what if you can just have the system do that on its own? It can monitor the cells, see that there's a certain number of them, engage a pump, and inject the dish with your drug. Maybe you're interested in finding a certain event. And whenever it finds one of those, it takes a Zstack. It's a pretty simple example, but it takes a long time if you do it manually, right? Maybe you're interested in detecting a certain state that your tissue or your cells are in. This state might manifest itself in different ways. Maybe the color of the protein changes, or you get expression of a different protein, a different channel, right? And then you want to do an uncaging experiment with a laser that uncages glutamate or some other cell signaling molecule. Or maybe you want to get as much data as you can before your cells die. This is actually more common than you think. Sometimes the limiting factor is just, well, I can only keep my cells alive or healthy for so long. We have technologies now available to keep cells alive for a pretty long time. But also, it's possible that the experiment that you're doing actually does kill the cells, right? And you want to collect data up until that point. So if you have a marker for cell death, you can continuously monitor that, or have the computer continuously monitor that, and then say, all right, once there's a certain amount of death in my sample, stop the experiment. It's sort of a little joke there because if you set it up a certain way and your computer is online, you can actually have it send you a text that it's finished, which is pretty cool too. Another example of something that you might want to be able to do is interface with an external device. We kind of touched on this a little with the injection of a drug, right? I know for a fact that a lot of Jim's microscopes have triggering, what we call triggering, and have NIDAC components, these PCIe chips here that go into the back of the microscope and allow you to send signals in and out. So one of the things that Elements is able to do is make an intelligent decision and then send a signal. Maybe that signal is a 5-volt signal that turns on a pump, for example. But it could be anything, really, anything that can be controlled by voltage. Maybe you have custom chambers, right? It's not necessarily that you want to do something super complicated, but you want to do that maybe something that's more basic, but you want to do it in a sample chamber that isn't typical. Well, Elements has the ability to allow you to define custom plates and custom weld plates, and then utilize those as easily as you would a more standard size plate or sample holder. So you see where we're getting at here, right? There's these tools in Elements that allow for this automation they're beneficial for folks who want to do really complicated things. They're beneficial for people who want to do really unique things. They're also really beneficial for people who want to do things that are more common, but they want to be able to do it faster, right? All of these things are possible. And, this is where I think people get excited. And then they're like, well, that's exciting. That sounds great. I think that I might be able to utilize these kind of things, but how easy is it to implement this? In the past, people have done things like this, but you kind of had to be a programmer in order to do it. You had to be able to interface or write a piece of software that can interface with these different hardware components and do it in a way that allowed them to all work together. It wasn't easy. So you've probably heard Jim talk about this because he's a huge fan of it. It comes down to something that we call jobs. And let me see, I think I could play this here. So what we've done, worked on my, oh, there we go. Okay. So what we did is, in order to balance the ease of having a graphical interface with the infinite possibilities of writing a program, of doing programming, for example, we created this user interface called Jobs. And the way that it works is on the left here, you have a list of tasks. You can kind of think them, if you are a programmer, you can kind of think of them as functions. And basically what you do is you just arrange those tasks in the order in which you want the things done. And it works similarly to programming in the sense that you can define stuff, like you could define what colors you want to image, which would be akin to defining a variable. You could define what plate you're using. Maybe it's the custom one, like we talked about a couple slides ago. Or it could be one of thousands that are in the database already. So you just select what model, what the manufacturer of your plate is, and it'll automatically load that in. And then you have what are called loops. This is really the main concept of what you need to understand. And it's used a lot in programming, but if you're not a programmer, it's totally fine. Just think about it this way. You have a lot of different things, like maybe you have a lot of different locations, or you have a bunch of different wells, 96 wells, right? For each one of those wells, you're going to loop through them. So you're going to go to well 1, well 2, well 3, well 4. And within that, you're going to do some kind of imaging. Maybe you're going to capture an image in the middle of the cell. That would be a simple example. Or maybe you want to take 10 random images throughout the cell in a non-biased way. You could do that too. And it just gets more and more complicated as you want. Takes a little bit of practice. I'm not going to lie to you and say right out of you're going to sit down and be able to do it, but you probably actually will. Once Jim or somebody shows you the basics of what you need in order to build a job, with a little bit of thought, it's all of you are more than capable and intelligent enough to be able to figure this out and do it, and probably come up with some pretty interesting and unique things that are really specific to your experiment. I really, really urge you to give it a try when you have some time, because that little bit of investment will save you a lot of time and allow you to do experiments that you didn't think you were possible to do, just because simply doing it manually would be too timely or too biased to do it. The other thing that we do is something called general analysis. This is an example of general analysis 2. It's a little bit more straightforward, but basically what it is it takes a lot of the individual analysis tools that are scattered throughout NIS-Elements and allows you to arrange them in a way that can be stored and reproduced. and also allow you to use them with each other. We've moved a lot to General Analysis 3, which uses, rather than having this graphic interface that is more traditional window with drop-down menus where you select stuff. Instead, it looks a little bit more like Jobs, where on the left side you have a bunch of different functions to choose from, and then you arrange them in these trees. So, for example, you have your different colors, and then maybe you want to do like a local contrasting on them, and maybe you want a threshold, and then you want to threshold on the cell, and then you want a threshold on these other little structures that you have here. And then you want to say, all right, how many of these little green dots are within a cell? How would you do this before you had General Analysis 3? Well, you would take a bunch of images, and then you'd look at them. And depending on what level you are in the lab, you would either do it yourself or hire an undergrad to do it and go through each one of these and count how many are within a cell, put it into a table, go on to the next image, and so on and so forth. And every single time you do it, you get a slightly different answer. It would take days, weeks, months. I know this because I did something like this, and it took me years to get my PhD. A lot of the time was counting things manually. I always say if I had NIS-Elements, I probably would have finished my PhD in half the time, because I wouldn't have had to do all the work that I did manually. So those are our two automated programs. We have Jobs, we have GA3. There's your table. See, you didn't even have to put it into a table, because the automated analysis spits it out. And remember, once this is built, you can apply it to as many images as you want. So if you took 100 or 1,000 or 5,000 of these images, you can apply this to every single one of them, and it'll use the same parameters and consistently count what you trained it to count in a really non-biased way. So let's talk about some examples. Let's combine these two things, Jobs and general analysis, and start to get a sense of the types of things that Elements is capable of doing with these new tools that we're talking about. Start with something simple. Let's make a job that takes Z-stacks at predefined points. So you go through the image, you find all the areas that you want to do a Z-stack, and so here we go. We're going to define the points. Let's see if I can make this a pointer here, a laser pointer. Here we go. So you define the points. Maybe at each one maybe you want to make sure that your Z-stack is going to have 3 different channels. So you say, okay, our capture definition is going to be DAPI, GFP, and Texas Red. Then we're going to go to each one of those points. Maybe we'll do a time lapse, and then at every nth time point, we're going to do a Z-stack. So for that Z-stack, we'll capture all 3 of these colors. And then when we're done with the Z-stack, we will measure the intensity of the green channel, and then put that into a chart. And then at the end, not only do we have the Z-stacks that we predefined earlier, we don't have to constantly stop and search for another one. We find them all. It does them. It does the time series. It does the Z-stack automatically, does the analysis, spits out the data. We can define the analysis here. We can say, usually you have to take a representative image first to train on. and then you can apply that to the automated experiment. So this would be a simple job and you see it's not that many steps, right? Pretty easy to do, and the logic is pretty clear. What points, what kind of images, go to the points, do a time lapse, do a Zstack, do an analysis. But what if you want to do something a little bit more complicated? Like, not only do I want to do this time lapse with Zstacks, but maybe I want to do this over and over again. And when I find a cell that has a certain green intensity, if the green intensity is greater than or equal to this value, then I want to do a fast time lapse on that. Now, I understand that this particular example might be meaningless to you and to your experiment, right? Maybe you don't care about doing Z-stacks and fast time lapses when the intensity is green, right? But that's not the point of this. The point is that this is just an example of what can be done. You can choose a bunch of cells, take images of them, measure on the fly some aspect of the structures that you're interested in, have it determine if you've passed a threshold that is meaningful to your data and to your experiments, the size of the cell, the intensity of a fluorophore, the shape of an object, and on and on and on. And when it meets those criteria, do something else. This is something that we do all the time, right? When we're doing microscopy experiments, we're trying to find the things that we're interested in. And then when we find them, we want to go to those and we want to do a time lapse, or we want to take a bunch of Z-stacks really quickly, or we want to add a drug and then do a time lapse. But a lot of times we end up going to random areas, getting a bunch of data that doesn't have anything interesting in it, or we spend all of our time trying to find the things that are interesting and don't actually get a lot of data. In this case, you can predetermine where a bunch of different points that you want, or tell it to find 100 or 1,000 random points or whatever, and do this, do the analysis on the fly. If it meets the criteria, do the experiment, move on to the next one. Jim and I were talking about this earlier. He actually made a 3-pass job, which is impressive. But in this case, maybe when we say 2-pass job, we mean go and take some images and then go back to something interesting and do another experiment. So here we're doing a large image, then we're going to revisit those locations and do a high magnification time lapse. So we're going to do the first pass with a low mag, then we're going to switch to a high mag, and we're going to do a time lapse. And at the same time, if there's a certain number of cells, we're going to perform the experiment. Otherwise, we're just going to move on to the next point, because I guess in this case, whoever wrote this job thought, well, if there aren't a certain number of cells in the field of view, then it's a waste of time to do the experiment. So don't waste that time. Just go to the next point and see if there's enough cells. And then if there is, do the experiment there, right? You see, our job is getting longer and longer here, the more complicated it gets. But the logic is still the same. Still really easy. We still have our points. We still have our capture definition. We've thrown in a cell count assay here, a pre-built one. And then we're just saying, hey, look at what we've counted. Go to those locations. If the cells are above a certain amount, do the experiment that we had been doing previously. If not, go to the next point. And now we have a situation where we have information across your entire sample. We know how many cells are everywhere. And then we have the experiment that we're really interested in only being done where it really matters. And you don't even have to be there to do it. You just have to be there the first time, make sure it works. It's a little bit more complicated than that. It takes a little while to get it to work, but it's really worth the investment. You can even make it easier on yourself. You can add wizardized steps. So for example, you might have some differences in your experiments. Sometimes you want to do 2 colors, sometimes you want to do 3 colors, maybe sometimes you want it to format the results in a different way. These kind of things can be done, can be wizardized, which means that when you run this job, it basically, before it runs it, it prompts you with a bunch of things to fill out, like, hey, what colors are you doing? Do you want a graph or do you not want a graph? That kind of thing. This is something that I like to touch on, and not so sure how applicable this is to you, but there is actually a development website. Jim might be aware of this, where you can interact, where you can actually talk to a wider community of people who use NIS-Elements, and use our microscopes to like interact with them with other programming languages and stuff and other external processes. And, I don't have too much experience with this, I'll just be honest, but we do have folks at Nikon who are really great with this and can help with outprocking, what we call outprocking to other already developed assays to like other programs. So if you have another program that can use the data from NIS-Elements and do something with it and then send it back, if you have an idea of something that you're working on where maybe this could work for you, definitely reach out to Jim and we can put you in touch with the folks at Nikon who really understand this stuff deeply and can help you work through that. I just wanted to throw that in at the end because it's interesting. If there's something that you can do in another program that isn't in Elements, you might be able to still work that into the job, which is pretty cool. So before I move on to AI, does anybody have any questions about jobs that pertains to maybe your own work, or you're just interested in other things that it can do? I'm happy to answer any questions about it.
I have a quick question.
Sure.
When you're talking about needing to train train the system before you can run a job, how much training data do you need? Is that an extensive step of needing to collect training data?
So it's less. So I say training is actually going to be more important when it comes to training an artificial intelligence network, which is what we're going to get into next, actually. But for the job, let's say you have a bunch of cells fixed on a cover slip, for example, simple example, and you're interested in cells that are the smaller of the cells. So then what you need to do is take a couple of images and use the sliders within the job where you would isolate small cells from big cells and make some determination where those parameters have to be so that it selects the cells that you're interested in. Otherwise, you still have to tell it what to look for. So that's what I mean by training. You're not really training it as much as you're optimizing the job and the analysis workflow for your particular data set.
Awesome. Thank you so much.
Yeah, no problem.
Joe, I have a question.
Okay.
So one of the tasks that I often do is kind of a whole slide scan.
Yeah.
And one of the bottlenecks is stitching that together because it's
Yeah.
such high resolution, just too many pixels for the local PC.
Yeah.
So I'm wondering if you've ever seen anyone use an outproc to like, upload that data to a cluster that can stitch it and then maybe downsample it and bring it back in for a GA3, for instance.
I haven't. It's possible, but what I usually suggest someone do is rather than capture a large image, is that you create a stage area and then tile that and then do the analysis on each individual image. It's much faster to do it that way. And then you can just tile it at the end if you want to have the tiled image. Or you could tile it after. Yeah, that's really what we suggest because there's so much, it's so intensive to tile, especially if you're doing like an entire slide, that it really becomes a time bottleneck. So, I mean, all a large image is a bunch of small images. So if you just keep it as the small images, then you can do the analysis faster. And you still, once the job is finished, it gives you that plate view. It kind of looks like a tiled image anyway. Like you know where the location of every image is. So that data is still there. Yeah. Cool. Anyone else? All right. So, I just want to talk a little bit about some of the artificial intelligence modules that we're working on and that are actually done now and are being used successfully. The idea here is that all the stuff that we're talking about, we're using our brains, right? We're looking at the data and we're saying, these are the cells that we know that these are the cells that we're interested in. We know that this is what we want to measure. And then when we try and play with different parameters on a computer to teach the computer, hey, like the software, hey, like we want to see this, or we want to focus on these structures, right? Notice it becomes really hard to explain to a computer what you want, right? And from when we're looking at it, it's very intuitive. We're very visual creatures, right? We're able to see the patterns easily that sometimes when you try and put it into a set of parameters that you teach a computer, you realize that what our brains are actually doing is pretty complicated. But what if you can use something like artificial intelligence, which is based on neural networks, in order to be able to accomplish what we do so easily, but can't do that fast, right? Before I get into it, because a lot of people ask this, what are our artificial intelligence networks based on? They're based on UNet. And then we work with them and do our own thing to basically accomplish the goals that we want these to accomplish. We don't really get into this because there's a lot of development on the backside on how this works. But if you're interested in the platform that it uses, this is the platform that we use. So let's talk about the general workflow. We talked about training, Madison, right? In this case, we're actually going to do training. We're going to create a data set that contains a ground truth that is already manually accomplishes what we want. Then we're going to provide those to the neural network. And we're going to say, hey, this is what we have. This is what we're going to give you. This is what we want. Create a classifier file that links the two. Then once we have that classifier, then we can apply it to just the first set of images and get the second. Some of these we've trained already. And some of them you can train for your specific data. It depends on which artificial intelligence network we're talking about. So let's go through some of them. One of them is called Denoise AI. This one essentially removes the shot noise in an image. Originally, it was for our resonance scanning confocals, but now it's more widely applicable even to the shot noise in cameras, for example. It's pre-trained on the distribution that noise is in a typical sample. And it allows you to basically capture images using parameters that will let you be more gentle and image faster at the expense of having a high signal to noise image, and then remove that noise afterwards and create an image or leave you with an image that is as good as one that you might have taken, but would require more energy in the sample and slower imaging parameters. So for those of you who have less experience with imaging, everything is give and take with microscopy. It's like life. You know, you do one thing, it helps you, and then, you know, maybe you want to get something else and you do it at the expense of another thing. So if you want to image fast, then you can image fast, but then you're getting less light because you're taking the images really fast and collecting less light, right? So you get a noisier image. It's the same thing. In this case, you know, maybe you want both. AI allows us to have both now. You can image fast, get a noisy image, and then use artificial intelligence to remove the noise and be left with an image that is just as good as one that would have taken more time to capture. Our new confocal, Jim, I don't remember if you have the AX yet.
Trying, not yet.
Not yet. All right. Well, when you have the AX, I know one of the things that you teach everybody when they come in to use the confocal is how to optimize parameters. What gain settings do you use? What laser settings do you use? How do you determine if an image is quantifiable? Are there undersaturated pixels? Are there oversaturated pixels? These are the kinds of things that make confocal imaging a little bit more complicated than other types of imaging. There's a few parameters that you have to learn how to use. It's not difficult, but you know, you're kind of working through it, figuring it out. Instead, you could just use this pre-trained artificial network that allows you to basically, you just click the button and it automatically chooses the optimal laser power and gain settings based on a few different inputs. So you might tell it, hey, I have live samples that are really sensitive. Put that setting in, hit the button, and it'll give you a laser power and gain setting that produces the best signal to noise possible based on those parameters. That also gives you an image with no undersaturated or oversaturated pixels. So it balances all the things that you're normally doing, but it does it really fast, and it's way more reproducible and perfect than a human being would be. It's pretty cool. I use it all the time now. I don't even have to, you know, when I find myself on confocals that don't have it, I'm like, oh yeah, I have to like do this all by eye and do some tests to make sure that my image is quantifiable, you know. One of the things, so those are both kind of really confocal based. One of them is more to optimize an image. The other one is to optimize an image afterwards, right? But what about for analysis? So this one called Convert.ai, really great tool. If you ever did phase imaging or DIC or anything like that, where you're just using white light and getting a contrasted image like this, we could look at this and say, yep, you know, here's a cell, here's a cell, here's a cell, here's a cell, right? But believe it or not, if you try and use typical thresholding tools that are used in image processing to tell a computer what's a cell and what isn't, it's almost impossible. It's not impossible, but it's almost impossible to do it using traditional methods, even though it's so easy for us to see it, right? And what's great about imaging cells like this using white light is that it doesn't really damage them. And you actually do get pretty good contrast. But what if you wanted to get some kind of information out of it? What you can do is use this convert.ai and predict a missing channel. So you can essentially get an image that would be as if you labeled it with a nuclear stain, but without actually doing so. The way that you do this really is you take images of in DIC or phase with cells that actually are labeled, and you train the network, and then you can do your experiments with just the unlabeled cells, and convert.ai will predict the position of where the DAPI label would be based on like super high dimensional things in the image that it determines using the neural network. So you label the cells, you train it, you have their classifier, then you can just use the cells that aren't labeled and get a labeled image. It's pretty powerful. And I think that things like this are going to be utilized more and more in circumstances where maybe having the label itself could affect the process that you're studying, right? We know that this is the case. Sometimes the thing that we need to do in order to see what we want is actually affecting the process that we're studying. So if you could train a neural network to do it after the fact and be able to utilize the power of fluorescent imaging without actually doing it, could be really powerful for your experiments. You could segment, you can do all the stuff that we talked about earlier in this talk, right? This is kind of in the similar vein as Denoise AI. You can take images really fast with really low signal to noise and restore the image with enhance that AI so that you can then segment it. It works in a similar way. What you would do is you would take a bunch of your images with fast parameters that you want to do your experiment with. Then you would do it with really like slow parameters and lots of power. So you get a high signal to noise image, something that might be damaging to your sample. But if you did it for long enough, but you do it enough, just on a few examples so that you could train the network, then you could do all your experiments with the fast parameters and get an image that allows you to, segment easily as you would on a sample that has high signal to noise. This would be the training and the implementation. So you train it on on your ground truth pairs, and then you can put in kind of a crappy low signal to noise image and get something that you can then segment more easily. And segmenting obviously is something that a lot of people do. A lot of neuroscientists, you're segmenting axons and dendrites. This is what I did when I was in graduate school. It took a long time. It was hard to find a program that can do it accurately. So I ended up doing a lot of it by hand. And this tool is really amazing. I've seen some examples of it, and it's just basically all you have to do is do it by hand on like a handful of images, train the network, and then it's extremely accurate on every subsequent image that you give it. And it doesn't even have to be fluorescent images. You could do DIC images, phase contrast images, anything that captures what you want to segment, and you can train it And it doesn't have to be branchy segmentation like this. It could be identifying a rare cell type amongst a bunch of other cell types. Maybe you have a dish with 2 different cell types in it, or a tissue that has multiple cell types in it, and every once in a while there's the cell that you're interested in. You know how to recognize it because you're an expert. Teaching a computer is really difficult. So what you do is you segment it manually on maybe 5 or 10 images, depends on your sample, right? And then you train the network, and now all you have to do is capture the images, and segment.ai will find all of those individual cases and segment it for you. Hand segment, train, get a classifier, implement. Now you have it. Clarify.ai, I think this is the last one we're going to talk about. This one basically, if you notice, if you ever taken images with a wide field microscope versus a confocal microscope, the wide field ones, especially if you're imaging something that's pretty thick, you get a lot of haze because there's a lot of out of focus light that isn't being removed like it is in a confocal. Clarify.ai has been trained to remove out of focus light and generate images with point spread functions that are similar to that you get from a confocal. And what's nice about it is that it works on images that have really low signal to noise, because traditionally what people would do is they would do deconvolution on a wide field image to sort of get this effect, which works really well, and it's quantifiable, right? But you need a certain level of signal to noise to do it. So if you don't have that, this can maybe be beneficial as well. Just to wrap this all in to what we talked about, we have, let's say we take general analysis, right? We talked about this earlier, these automated analysis workflows. You can incorporate your trained neural network, your AI classifiers, into general analysis. So not only are you doing typical analyses within general analysis, but you can also apply your classifiers as well. You can use that for real-time analysis within a job. Maybe now that you're able to quickly and accurately identify structures in your sample that you weren't able to do as quickly and as accurately on the fly, now that you have a classifier that can do it, now you can incorporate that into a job and have the job make a decision based on that, and do it, and actually be able to accomplish experiments that were impossible to do before, simply because you couldn't analyze images accurately and fast enough in order to make an on-the-fly decision. So I think that's really where AI is going to have a huge impact on the ability to capture lots of data and good quality data that's useful. That's really where it's going to, I think, have a huge impact. So, all of this, remember, we kind of focused mostly on the analysis side and the automation side, but I just want to, in the last slide or two, discuss and point to the fact that this isn't limited to one of the microscopes in Jim's lab, in his core facility, in the COE. This is applicable to a lot of our hardware and our microscopes, inverted and upright microscopes, point scanning confocals, spinning disk confocals, and many other imaging techniques, TURF, multiphoton, FRAP. I know last time I was there, someone was doing FRET. And, doesn't matter what you're doing or even what imaging technique you're using. These tools like Jobs, General Analysis, the NIS.AI, neural networks, all of these can be utilized with these different techniques and these different microscopes. And so with that, I just wanted to put up your Nikon team and open it up for questions. I think our timing was pretty good. Went a little over. Sorry, Jim.
It's all good, Joe. That was really great. So if anyone has questions, feel free to pick Joe's brain here. And if anyone has questions about, the offerings that we have, you can just email me. Like Joe said, we have jobs on, I think, 6 or 7 of the microscopes. So at the very least, each modality, wide field, spinning disk confocal, and scanning confocal, as well as some of the other scanning confocals. And we have GA3 also on those, as well as offline workstations, including the AI modules and deconvolution and stuff like that. So I think pretty much everything you talked about, we have, here today. You could use it. 5 minutes. So that was awesome, Joe. So Joe, thank you, and thank Nikon for giving us the time to.
Of course, love it. And
experts.
I'll give everybody one last chance to ask a question. I'm easygoing. Don't be shy. No questions are dumb. It's all good.
It's true. There's no dumb questions in microscopy.
There isn't. It's all, learning and practice, and you could do amazing stuff.
Yeah. Well,
all right.
Thank Lisa and Andrew for hosting again. So
yeah.
All right.
Thank you both.
All right.